Hi, in this video, we'll be talking about several antibodies isotypes. Now, isotypes are basically subclassification of antibodies based on their heavy chain and the light chains. Now, we are talking about several classes of antibodies such as IgG, IgE, IgM, IgA, etc. So in this video, we won't be kind of focusing on the structural aspect of it. Yeah, of course, the structural aspect is important and that reflects to the function of it. But in this video, we will be mostly looking at what antibodies are most abundant in our body, what are their functions, and how they are biologically relevant. First, talk about IgG. So if we talk about all the antibodies, IgG is almost 75% of all of that. So that makes it the most abundant antibody in our body. Importantly enough, IgG can cross placental barrier and it can go to the newborn. So when the baby is developing inside the mother's womb, its immune system is not mature. Its immune system is still forming. At that situation, mother derived IgG is the source of its immunity. So that is why IgG is very important. Other than these functions, IgG can also fix complement and also take part in complement mediated uh, attenuation processes. So in that situation, IgG is super important, and its complement fixation capability is pretty high. Next, we talk about IgM. Now, in terms of molecular weight, IgM is the bulkier one, and also the membrane-bound B cell receptor is basically IgM. Now, but the secreted IgM is basically pentameric and kind of a oligomer of these membrane-bound monomers. Now the question comes, why some secreted version is like a pentameric whereas a membrane bound version is monomeric. It turns out the difference between the secreted antibody and the membrane bound antibody is in the transmembrane domain. In the transmembrane domain is only present in the membrane bound form, which is basically B cell receptor. But when the B cell gets differentiated into plasma cell and secrete the antibody, the secreted IgM don't have this membrane bound domain, so that's why it can be easily secreted, don't get tethered to the membrane, and also with other J chains it forms a pentameric structure. Now IgM is also very important in terms of complement fixation. It has huge potential of complement fixation because it has so many uh, paratopes, right? Now IgM level spikes just after antigen exposure. I mean, in fact, the IgM is the first antibody to be produced after any primary antigen exposure. So that is why IgM is also very important after IgG. Next, we talk about IgA. IgA is basically a dimeric kind of structure. You can imagine two monomers are linked together by a J chain, and it is mostly found in secreted fluids, and it can exist as dimer mostly, but also it can exist as tetramer. Now IgA can be found in secreted uh, fluids like breast milk, saliva, tears, mucus, etc. So we just try to understand the process of this process and by an example of transcytosis. So transcytosis is a process by which several protein can get endocytosed from one side and get transported and exocytosed from other side, and that is how it can cross a cellular barrier. So you can understand in breast milk the IgA antibody is basically secreted. So how does it happen? So if we look at the milk ducts, so we would see there are epithelial cells and the lumen. So what happens is the plasma cells secrete antibodies which are IgA in nature, and they kind of go and bind to poly-IgR receptors on the epithelial cells. Now poly-Ig receptor binding to the IgA leads to its receptor mediated endocytosis. As a result, it gets internalized, and from the other side it can be exocytosed and released into the lumen, I mean where the milk is actually getting produced. and with that it can be secreted in the environment, right? Now coming to IgE level in the blood is the lowest. So it's like constituting like 3% of all the possible isotopes found in human body. Now normally IgE level is less, but some people have abruptly high IgE levels, and those people are termed as atopic, and they are like sensitive to allergic responses. So in a moment we would see what is the role of IgE in evoking allergy. So it turns out, let's say this human is getting affected by the pollen grains. So when the pollen grain enters his nasal mucosa, and the dendritic cell or the macrophages encounter the pollen grain, they would eventually allow activation of the T cells, and these T cells would eventually allow the activation of the B cells nearby. Now, these B cells would differentiate soon to become plasma cells. Now, the plasma cells would secrete IgE. By that time, the mast cells are also getting primed. Now, the mast cells has IgE receptor. So, whenever a second time the individual is getting exposed to the allergen, the IgE produced that time binds to the IgE receptor of the mast cells and allow the mast cells to be sensitized and degranulate in the second time, right? And degranulation of the mast cells cause smooth muscle contraction, vasodilation, bronchoconstriction, and glandular secretion, and that's how the all allergic symptoms, inflammation, pain, it evokes. So I hope you enjoyed this video, and in this video we described all the subtypes of the antibodies and their functions, their abundance in our body. So if you like this video, give it a quick thumbs up. Don't forget to like, share, and subscribe. Thank you.